Hello there, and welcome to Smart Student TV, the only channel where academic and professional prowess is nurtured right from the comfort of your living room. So, whether you are a student striving for excellence, a professional looking to sharpen your skills, or simply a lifelong learner, you are in the right place. In today's tutorial, we dive into another very crucial topic: sterilization and disinfection. Before we dive into today's topic, let's do a quick recap. In our last session, we explored laboratory cleanliness. Now, let me ask you a question. Why do you think a clean lab is essential? Well, cleanliness in the lab isn't just about looking tidy. It's about safety, accuracy, and professionalism. We discussed how a clean lab prevents cross-contamination, avoids chemical hazards, and ensures reliable experimental results. Now here's the connection. Cleanliness is just the first step. Think of it like washing vegetables before cooking. Cleaning removes visible dirt, but sterilization and disinfection ensure that even the invisible threats, like bacteria and spores, are dealt with. So while cleanliness makes the lab look ready, sterilization and disinfection make it safe. In simple terms, cleaning removes visible dirt, dust, and contaminants, whereas sterilization eliminates all forms of microbial life, including spores. On the other hand, disinfection reduces microbial presence to safe levels. Here is a quick example. Wiping a lab bench with water is cleaning, but wiping it with a disinfectant ensures harmful microbes are minimized. Objectives of today's tutorial. By the end of this session, you will 1. Understand key terms related to sterilization and disinfection. 2. Differentiate between cleaning, disinfection, and sterilization. 3. Explain the importance of sterilization and disinfection in laboratories. 4. Identify common reasons for incorrect sterilization practices. 5. Describe methods of sterilization, their applications and limitations. Importance and significance of sterilization and disinfection. Why do we care so much about sterilization and disinfection? Let me paint a picture. Imagine a microbiologist working with dangerous bacteria. Without sterilization, any leftover bacteria could spread, causing infections or even outbreaks. Benefits of proper sterilization and disinfection. 1. Protects lab personnel from infections. Proper sterilization ensures that harmful microorganisms are eliminated, reducing the risk of infections that could compromise the health of lab staff. 2. Prevents cross-contamination between samples. Effective sterilization prevents microbes from transferring between different samples, ensuring the integrity and reliability of laboratory results. 3. Ensures accuracy of experimental results. Sterilization minimizes the risk of microbial interference in experiments, leading to accurate and reproducible results. 4. Complies with safety regulations. Laboratories are required to adhere to strict sterilization protocols to meet safety standards and avoid legal or regulatory consequences. Consequences of poor sterilization. So, what happens when we neglect proper sterilization and disinfection? Any or all of the following can happen. 1. Increased risk of laboratory-acquired infections. Poor sterilization can leave harmful microorganisms on equipment and surfaces, posing a significant health risk to lab personnel. 2. Invalid or compromised experimental results. Residual contaminants can interfere with experiments, leading to unreliable or incorrect outcomes. 3. Spread of infectious diseases. Improper sterilization can result in the spread of pathogens beyond the lab, potentially causing larger public health concerns. 4. Damage to expensive lab equipment. Residual contaminants can corrode or damage sensitive laboratory equipment, leading to costly repairs or replacements. 5. Legal and regulatory consequences. Failing to meet sterilization standards can result in legal penalties, fines, or even the closure of laboratory facilities. So, why do some technicians still miss the mark? Let's discuss. 1. Lack of proper training. Many technicians do not receive adequate training on sterilization techniques. This can lead to incorrect procedures, improper handling of sterilization equipment, and ineffective outcomes. 2. Time constraints. In busy laboratories, technicians are often under pressure to complete tasks quickly. This can lead to shortcuts being taken during sterilization procedures, reducing their effectiveness. 3. Improper use of equipment. Sterilization equipment such as autoclaves and hot air ovens require precise operation. Misuse or failure to follow standard operating procedures can compromise the sterilization process. 4. Negligence. Sometimes, technicians may underestimate the importance of thorough sterilization, assuming that basic cleaning is sufficient. 5. Limited resources. Some laboratories may lack access to proper sterilization equipment, disinfectants, or maintenance services, making it challenging to perform effective sterilization. Each of these factors highlights the need for continuous training, better resource allocation, and stricter adherence to sterilization protocols to ensure laboratory safety and efficiency. Terms used in sterilization. 1. Sterilization. Destruction of all microbial life. 2. Disinfectants. Chemicals used to eliminate microbes on surfaces. 3. Antiseptic. Chemicals used on living tissue to reduce microbes. 4. Bactericide. Agents that kill bacteria. 5. Bacteriostatics. Agents that inhibit bacterial growth. 6. Biotoxin. Toxic substances from microbes. Sterilization methods. Sterilization methods are primarily divided into two major categories. 1. Physical methods of sterilization. These involve physical agents like heat, radiation, and filtration. 2. Chemical methods of sterilization. These rely on chemical agents called disinfectants such as alcohols, aldehydes, and gaseous sterilants. Both approaches have unique strengths and are chosen based on the material, level of sterility required, and the application. 1. Physical methods of sterilization. Physical sterilization methods use tangible factors like temperature, light, or physical barriers to destroy or remove microorganisms. These methods are categorized into heat methods, radiation methods, and filtration methods. Let's start by discussing the heat method. Heat is one of the oldest and most reliable sterilization methods. It works by denaturing proteins, disrupting cellular structures, and oxidizing essential components in microorganisms. Heat sterilization is divided into two major types. Dry heat sterilization. Dry heat sterilization uses hot air without moisture to kill microorganisms. Dry heat method can further be categorized into 1. Red heat method. In this method, sterilization is done by holding materials in a Bunsen burner flame until they become red hot. This method is commonly used in microbiology labs to sterilize inoculating loops. The method is quick and effective. However, it is only suitable for metal instruments. 2. Flaming method. Flaming method is used to prevent microbial contaminants from entering a sample. For example, to transfer a culture from an agar plate to a tube, you can flame the inoculating loop or mouth of media containing tube or bottle, then let it cool for 15 to 30 seconds before using it. This method is used for sterilizing small glass tools. The method is simple and efficient. However, this method cannot sterilize heat-sensitive tools. 3. Hot air oven method. In this method, tools are heated at 160 to 180 degrees Celsius for 1 to 2 hours. This method is often used to sterilize glassware and metal tools. This method leaves no toxic residue on the surface of the items. However, the method is time-consuming and unsuitable for heat-sensitive materials. 4. Incineration method. In this method, waste materials are burned at extremely high temperatures. Incineration method is often preferred as a safe disposal of medical waste. The methods completely eliminates pathogens. Nonetheless, this method raises environmental concerns due to emissions. 2. Moist heat sterilization. Moist heat sterilization is more effective than dry heat due to better heat transfer and the coagulation of proteins. It uses hot liquid or high-pressure steam to kill bacteria, viruses, and spores. Moist heat sterilization can further be categorized into 1. Boiling method. In this method, items are boiled in water at 100 degrees Celsius for 15 to 30 minutes. This method is often used for sterilizing surgical tools. It is a simple and low-cost method of sterilization. However, it is ineffective against bacterial spores. 2. Pasteurization. It is a method in which liquids are heated at 63 degrees Celsius for 30 minutes or 72 degrees Celsius for 15 seconds. This method is frequently used in preservation of milk and fruit juices. The method retains nutritional value of preserved food. It, however, does not kill spores. 3. Tyndallization. This method involves repeated cycles of heating at 100 degrees Celsius over 3 days. It is a method often used for sterilizing heat label culture media. It is effective against spores. However, this method is time-consuming. De-autoclaving. Autoclaving is a widely used moist heat method of sterilization that employs high-pressure steam to kill microorganisms, including bacteria, viruses, fungi, and spores. It is particularly effective for sterilizing equipment, glassware, liquids, and culture media in microbiology, medical, and laboratory settings. Autoclaving works on the principle of using high-pressure steam, usually at 121 degrees Celsius under pressure, 15 to 30 psi, to destroy microorganisms. The elevated pressure ensures that the water remains in liquid form at higher temperatures, allowing for more effective sterilization. Components of an autoclave. The main components include 1. Pressure chamber. A robust, airtight container designed to withstand high pressure and temperature. 2. Steam generator. Produces steam required for sterilization. 3. Pressure and temperature gauges. Monitor internal pressure and temperature to ensure proper sterilization conditions. 4. Safety valve. Prevents excessive pressure buildup by releasing steam if necessary. 5. Exhaust valve. Controls the release of steam after sterilization. 6. Sterilization basket/rack. Holds materials to be sterilized. 7. Door locking mechanism. Ensures the chamber remains sealed during operation. 8. Control panel. Allows users to set temperature, pressure, and sterilization time. Uses of an autoclave. An autoclave can be used for the following purposes. 1. Sterilization of laboratory glassware, instruments, and culture media. 2. Decontamination of biohazardous waste. 3. Sterilization of surgical and medical tools. 4. Preparation of sterile materials for microbiological experiments. Procedure for using an autoclave. Step 1. Preparation of materials. Place items to be sterilized in autoclavable bags or containers. Ensure containers are loosely closed to allow steam penetration. Avoid overloading to allow free steam circulation. Step 2. Loading the autoclave. Place materials in the sterilization basket. Ensure glassware and containers are not tightly sealed. Close and lock the autoclave door securely. Step 3. Setting sterilization parameters. Set the temperature, typically 121 degrees Celsius. Set the pressure, 15 psi is standard. Set the time, usually 15 to 30 minutes, depending on the load and materials. Step 4. Start the cycle. Activate the autoclave cycle. Monitor temperature and pressure gauges. Wait until the cycle is complete and the pressure returns to normal. Step 5. Unloading the autoclave. Allow the autoclave to cool before opening. Open the door slightly to release residual steam. Carefully remove sterilized materials using heat-resistant gloves. Testing the endpoint of sterilization. Ensuring sterilization has been effective is critical. Common methods used for testing whether complete sterilization has been achieved or not include 1. Biological indicators. Use heat-resistant spores, e.g., Geobacillus styrothermophilus, to test sterilization efficiency. If spores are killed, sterilization was successful. 2. Chemical indicators. Autoclave tapes or strips change color when exposed to proper sterilization conditions. 3. Physical indicators. Monitor temperature and pressure gauges during the cycle to confirm correct parameters were reached. All along, we've been discussing the physical methods of sterilization. Next, let's shift gears and dive into something super important and fascinating. Chemical methods of sterilization. Now, I know sterilization might sound like one of those intimidating lab topics, but trust me, by the end of this session, you'll not only understand it, you might even enjoy it. So, grab your notebook, Sit back, and let's get into it. 1. What exactly is chemical sterilization? Alright, let's start with the basics. Chemical sterilization is all about using chemical agents to eliminate or kill all forms of microorganisms, including those stubborn spores. Think of it as giving lab equipment or surfaces a deep chemical bath to ensure they're squeaky clean and microbe-free. You might be wondering, why not just use heat or radiation? Good question. Some materials, like plastic lab equipment or heat-sensitive medical tools, can't handle high temperatures. That's where chemical sterilization comes in as the hero. 2. Why is chemical sterilization so important? Now, let's talk about why this matters. Imagine running an experiment or performing a medical procedure with contaminated tools. Definitely, the risks are endless. 1. Contaminated results. 2. Spread of diseases. 3. Failed experiments. 4. Health and safety violations. In short, chemical sterilization isn't just about cleanliness. It's about accuracy, safety, and professionalism. 3. What are we trying to achieve with chemical sterilization? At its core, chemical sterilization has some clear goals. 1. Eliminate all microorganisms, even the stubborn spores. 2. Prevent microbial contamination. 3. Sterilize heat-sensitive materials. 4. Keep lab and healthcare spaces safe and compliant. What makes a good disinfectant? Here's the million-dollar question. What qualities should a good disinfectant have? Well, it's not just about smelling strong or bubbling on contact. 1. Broad-spectrum activity. It should kill a wide variety of microbes. 2. Fast-acting. Nobody has time for a disinfectant that takes hours. 3. Non-toxic. Safe for humans and animals. 4. Non-corrosive. Shouldn't ruin expensive lab equipment. 5. Stable. Shouldn't lose effectiveness over time. 6. Easy to use. Simple to prepare and to apply. 7. Affordable. Cost matters, especially in large-scale labs. 8. Environmentally friendly. Safe disposal is key. Let's talk about some popular disinfectants. Here comes the fun part. Let's break down some commonly used disinfectants and chat about their strengths and weaknesses. 1. Ethylene oxide, EO. It's a colorless gas with excellent penetration. Ethylene oxide is perfect for sterilizing heat-sensitive tools. However, this disinfectant is highly toxic and flammable. Not something you casually handle. 2. Formaldehyde. Formaldehyde is strong-smelling gas with powerful antimicrobial activity. It is great for sterilizing large spaces. Formaldehyde is, however, very carcinogenic and irritating to skin and eyes. Handle with care. 3. Glutaraldehyde. Glutaraldehyde is non-corrosive and broad-spectrum. It is used for endoscopes and other heat-sensitive tools. It is, however, a skin irritant and pose respiratory hazards to users. 4. Hydrogen peroxide. Hydrogen peroxide is a powerful oxidizing agent. It is often used for wound cleaning and surface sterilization. However, hydrogen peroxide decomposes in light, so proper storage matters. 5. Peracetic acid. Peracetic acid is effective against spores, viruses, and bacteria. It is commonly used in the food industry. However, it is highly corrosive. 6. Alcohol, ethanol, and isopropanol. Alcohol is quick-acting disinfectant most commonly used as a hand sanitizer and for surface cleaning in many facilities, usually at a 70% concentration. However, alcohol doesn't kill spores. 7. Chlorine compounds. Chlorine compounds are broad-spectrum activity disinfectants mostly used in water treatment. Chlorine is, however, very corrosive. 8. Phenolic compounds. Most phenolic compounds are stable and effective disinfectants mostly used for cleaning hospital surfaces. However, they irritate skin. What affects the effectiveness of disinfectants? Not all disinfectants work the same in every scenario. Here are the key factors. 1. Concentration. 2 diluted equals effective. but too strong equals very dangerous. 2. Contact time. The longer it is exposure equals better results. 3. Temperature. Warmer temps often speed up action. 4. pH. Some chemicals work better in acidic or basic conditions. 5. Organic matter. Any presence of dirt or blood around the surface to be disinfected can neutralize disinfectants. 6. Microorganism type. Bacteria with spores are tough to kill. 7. Surface condition. Any presence of dirt or blood on the surface affects its ability to disinfect, and therefore pre-cleaning helps. 8. Water quality. Hard water reduces efficacy. And there you have it. Chemical sterilization isn't just about pouring some strong-smelling liquid over tools and calling it a day. It's a science, an art, and a critical safety practice. So far, we've looked at 2 methods of sterilization, the physical and the chemical methods of sterilization. I think it's now time we look at the remaining 2 methods, namely, radiation sterilization and filtration sterilization. Let's begin with sterilization by radiation. Radiation sterilization involves using high-energy rays to kill or inactivate microorganisms. It's especially useful for heat-sensitive products. Types of radiation used in sterilization. Radiation sterilization can be categorized into 2: ionization radiation and non-ionization radiation. Ionizing radiation. Ionizing radiation is a type of short-wavelength, high-intensity radiation that can break the bonds of molecules and remove electrons from atoms, which can damage the DNA or RNA of a microorganism and kill it. These forms of radiation include gamma rays. These are like invisible bullets fired at microorganisms, damaging their DNA and preventing reproduction. For instance, disposable surgical gloves are often sterilized using gamma radiation.